Yes, I'm Leonid Savchenko. I'm from UCL Institute of Neurology, and I work more like a computational neuroscience. And today I like to present our new platform, computational platform. It's not a model. It's really like a platform. You can design 3D brain cell. And not just design one just with all of the details, nanostructure, all the mechanism. You can design many cells and put all together in like 3D space and see how they communicate each other. Like one release neurotransmitter, another uptake neurotransmitter. It means this platform allows just non-trained users, like a clinical scientists, biological scientists, neuroscience, create really very complex cell, design experiment, and put all together in one 3D space. Okay. Why brain cell? First, you see, when I start neuroscience job, my experiment was like a one-patch clamp, one cell, one setup. And we published good paper. But a few weeks ago, we submit paper in Science. It's like a 5 completely different methods, from imaging, FRAP, FLIM, I don't know, just genetic manipulation, 10 people. And each in vivo, in each experiment costs a lot of money. And normally, before you start an experiment, you need to just predict what you're expecting for. But brain cell exactly for this. You can design very complex cell, put together, and see how communicate neuron or different neuron or neuron with astroglia, and what your effect expect, and design experiment. Then you can design, measure concentration, extracellular, intracellular, measure voltage, and also you can measure extracellular field. You can just put all neurons together, they fire, and you just put electrode and see what's happening. Because neuron can fire in some frequency, extracellular phase, change resistance in another frequency. Okay, it's what I just told, it's designed for non-trained user. It means you don't need just to be very professional IT specialist. You need just very simple in IT, because using the new lingua model, you can, this is designed like a block. You can just very easily design your own channels, your own receptors, and add very easy to this platform. And this allows to accelerate hypotheses, predict, experimental, and so on. It means before starting very complex experiment, you can just design your own setup. Okay, but before I go just about brain cell, I invite you to our neuralgebra.net. It's a place where you can just very simple, this one, you can very simple upload 3 platforms, Astra, Arachne, is the new one, brain cell. Astra, it's an old one, but using the Astra, you can design any glial cell from nanostructure to very complex behavior, like a calcium vein, calcium dynamic, just diffusion inside the cell. But this is an old one, and after that we designed Arachne. Arachne, you can design networks. Not like an artificial network with like a Hopfield network or frequency network or spiky network. It's a real network. You can just add here Hodgkin-Huxley or another whatever you want mechanism, and you simultaneously design at least 3 communicated networks. Like a pyramidal cell, is just like example. Interneuron, and between them, astroglia. And you can see how they communicate, how design synchronization, and also all of this network is just for non-trained user. You can just, here very simple, but only for Windows. very great because you can just special code. You can just execute, install, and you have just panel, design parameters, synaptic communication, how many neurons, and so on, and then see many dynamics. And what is really important, you can put electrode around this network somewhere and see what's the recording like from the skull. Okay, and this is you can just go download. This is most simplest way, download and install, but only for Windows. And I today will just discuss about BRAINCELL. It's a new platform. Its platform allows you to create neuron like this, with all of these spines, synapses and so on, or neuron like this, with all of this detailed GABA release, diffusion and so on. Not only like typical model, diffusion inside cell. You can just study diffusion outside, I will show. Okay, this is main windows. It looks like a huge, but normally it's like 100 of different windows. And design for these windows, came from Neuron. Normally, BRAINCELL designed for 3 languages, most basic like Python, but we can also introduce a lot of neuron models. Why we still using Neuron? It's because a lot of database for neuron simulation. Like a database for upload shape, like this. I will show how easy BRAINCELL allows to upload different shape of different brain cell. Also, you have this huge database of any biophysical mechanism, like channels, receptors, and so on. When you go on neuron database, you can take the BIM mechanism, just say, I need sodium channels. You have this huge list of sodium channels. You copy, put there, it immediately appear in your model. It's very easy, and this is why still using Neuron. Okay. Very short, what is it new in advance in BRAINCELL? You can just design stochastic morphology. You can very easy upload shape like a pyramidal neuron from special database. Then BRAINCELL allow you to create like a spines or varicosities everywhere, or you design where and how many, what's the density and so on. And then what you can do, put synapses for them here. And you have windows. You can just manipulate distributions of synapses, distributes type of synapses, and so on, and see how change the response of this neuron, depending, for example, of release probability. But just as an example for neuron, you can do it, for example, astroglia. You can very easily create very complex astroglia and study, for example, dynamic of voltage in this astroglia. At the same time, you can manipulate the shape, distributions, density, surface, and so on, and see how it changes the voltage distributions. Okay, next. Normally, before, when you people study simulate some cell, everything happens inside cell. Voltage, membrane, diffusion. Because you see, you have like a basket cell. You download basket cell, and you need just how the GABA spread around the axon of basket cell here. You see, you need just to create special space, special matrix here, and you see this is a very complex. And we solve these problems. And this, you can very easily upload this basket cell, put a lot of GABA release site. Then you can just set how many GABA molecules release, how fast, what the synchronization, what time, and so on, just like a table. And then what you can see, you can just see dynamic of GABA in different way. You have just windows. Okay, I need just to understand how GABA diffuse. You see, this is a movie. Diffuse around the branches of axon during some times. Okay, but BRAINCELL allows you not just to see how it diffuse close to the axon, but how it diffuse in extracellular space. It's just another windows. And you can just create a movie and see, for example, if synchronous release, how fast it's uptake, because you can design extracellular space. You design diffusion coefficient, tortuosity, uptake, and so on. And you will see dynamic of GABA, just very simple and very easy. Okay, what is it? Something new? Again, typical when you study model of any cell, you assume the extracellular field is constant. We just only think about extracellular space when you just simulate. What's the extracellular space? It's electrolyte. A lot of like a solution of ions moving everywhere. When you stimulate, it's normally you just change, redistribute ions inside the brain. Not just specific electrical signal, because everything completely neutral there. Now, brain inside the soup of electrolyte, everything completely. When you stimulate, it changes. But brain cell allow what you can do. You design very simply nice astrocyte. It's human astrocyte. And then, let's look here. In sphere, release extracellular potassium here. Like normally concentration 2 millimole, you just increase 5. And see what's happened. Immediately you see how it changed voltage. Immediately you see how it changed intracellular potassium. It's example. You can just modulate any ions in extracellular space. Any. You just already in brain cell, like all main ions, sodium, potassium, calcium, so on, all neurotransmitter, GABA, glutamate, acetyl halide, and so on. And also you can just design your personal neuron. For example, your growth factor. You can just design, you have special tape, and then just release growth factor and see what's happened with dynamic of cell. Okay? But this is interesting. He says, but this is artificial. But what really interesting can you do in brain cell here? You can design pyramidal cell. Also you can design astrocyte. And put all together. And say, okay, pyramidal cells start firing, like a typical firing of pyramidal cell. And astrocyte doing their job. Just work like a sponge. Take potassium from the extracellular space. After 10 seconds, you will see a lot of potassium where the neuron firing, and around the astrocyte that's keep constant. But you can not just put only one astrocyte, you can put hundreds of astrocytes, depending on your power of computation. And you see how you need many astrocytes around pyramidal cell to keep constantly firing. Because sometimes people just concentrate, okay, you see you change the firing rate of neuron, but not because something inside, it's normally something outside. It's like in epilepsy. You just firing neuron, potassium going up, excitability growing up, and just block. In this way going across the brain. Okay, what is it? Also something really new. When people simulate, okay, I want to stimulate myelinated axon. And normally in brain, it's not my student, PhD student, normally see it's like a one cell. But normally it's not one cell, it's two cells. Neuron and Schwann cell. It's too difficult, two different cells. Why it's important to have simulate two different cells? Normally people just simulate, just axon, non-uniform distribution of sodium potassium channel, Randier node, and so on, and special parameter which looks like a control concentration. But when in some disease, in Schwann cell, some channels block or disappear, you cannot just simulate in this way, because Schwann cell communicate with axon only via extracellular space. It means you need just create really two cells, one cell, another cell, not extracellular, between cell communication in ions, and you can just see many windows. These windows show what's happening in different lines with potassium, close to the axon somewhere here. You just check potassium and so on. and you can just immediately control all the parameters. You can modify like conductance or some transporters of glia cell and see what's happened in some kind of disease. Okay, and very short here. Normally, everybody know, you're doing some simulation, if somebody doing simulation, you design very complex model, and then submit to Neuron, or just come next day. Okay, after half year, you completely forget all the parameters set up, but what you can do in BRAINCELL, you're doing something. Today you, okay, I'm tired, just save everything you're doing in one thin file. It's file, you can just walk or with BRAINCELL or separate. It just save all parameters. It means when you submit some paper, when you come for review, you can know exactly what you see. You're not afraid, oh, I forget, what does the parameter happen, what detail, but you know exactly everything because you just save here. And this is really important part. And you can just put different non-synaptic communication between cell. You can just add what I discussed, a lot of mechanisms, biophysical mechanisms for this cell. You can design, for example, I just only try, I take some paper and ask ChatGPT, can you create a mod file for BRAINCELL? It just create, I just correct in some way, and yeah, just add here. It's from published paper for a biophysical model here, like a 10 minutes now. It's still like a lingua model can just eat. I try to that lingua will eat this BRAINCELL. No, it's still not enough memory. Here it's roughly like a 500 files, too many for them still, but maybe in 2 years, I don't know. And you have just a lot of database for shape. Okay, it's very, I still have time, and here example of realistic astroglia. Okay, you go and experiment. You just record. You can just image and record shape and calcium dynamic. In Alexa, you can record this is beautiful astroglia. And another channel, you record beautiful calcium dynamic. Okay, this experiment very expensive. And you decide, okay, how is the calcium waves or calcium propagation depend on calcium buffer? If you can do an experiment, again, a lot of job, and you can predict. But what you can do with BRAINCELL, you can just create your own cell. First, you can recognize, or you can use specific statistic inside the BRAINCELL about nanostructure of astroglia. Nanostructure, it's like this. It's like a sponge. It's very complex, but we did for some astroglia create a specific statistic and allow you to create spongy form around dendrites and soma. Then, what you can do, this is your record astroglia here. Then you can very easy reconstruct main branches. And then you can just create numerical format. Now you have nanostructure, main branches, and what you next in the end, you have just combined all together. These are main branches. This is your nanostructure. And this is your final astroglia. And now you can just simulate, you see, nice calcium way. And you can just test different buffer, different concentration, membrane properties, and see how they affect calcium weight. After that, you can go and do new experiment. Okay, but you can say, okay, I need just reconstruct, I need just upload. No. In other options, you can go here, neuromorph.org. Here, 40 million shape of different brain cells. And you can go here, just design other Astra. You can find astrocyte. You can choose whatever you want here. You see, this is a, then just save all format, just all. You don't think, they propose like a 5, you don't think, just say all. Save in your brain cell, and somewhere here. Then go in brain cell and just upload this, and immediately you have in brain cell, immediately you have this astrocyte you already download from this database with all nanostructure, because we have a statistic here. It means from web page to astroglia, 5–10 minutes. And you can just start whatever you want. But you can just say, okay, we have shape, nanoshape. Now we need mechanism. It's very easy. Here we have special place, you can just put mechanism. You have just special for astroglia, for neuron, for another cell, it's called common, you can just put there. If you go, for example, for neuron, you will see like this, and you have special mod file, and this is file for build. What you can just do, copy somewhere mod file and put here. Then you just press here, build mechanism, and what's happen next? You go to brain cell, and all of this mechanism appear here. For example, it's a lot mechanism here, and what you can just do, you can just very simply click. For example, this example of neuron. In like a brain cell proposed only 5 versions: soma, dendrite, axon, spine-neck, and spine-head. But what you can do, in your case, you have 2 types of dendrites, like distal and proximal part. It's very easy. You can go here and split 1 into 2. You can appear windows and say, this distal dendrite, this is proxy dendrite. And you immediately here appear not 1 dendrite, will be 2 type of dendrites. And now you can just put different mechanisms there. Okay?
Can I ask
two
questions? The first question I would like to ask, I saw these ion channels. My first question is, can you change the kinetics? Okay, my other question is, did you guys take care of the units? Do they match the units? Because some parameters might be in, like, let's say, concentrations.
No problem.
Micromolar versus millimolar.
Absolutely. As soon as you put their mod file, doesn't matter, and just create a special add, they will check all this
discrepancies.
The discrepancy, and they say, okay, you need just add something. Okay. This is a very good. This is why I still use neuron, because they allow to do like this. Immediately, you need just to multiply 2 times because it's discrepancy. Okay. And okay. But when you add mechanism, for example, sodium channels. Sodium channels, you have just kinetics, parameters, you have special table for any mechanism. And for example, what you can do here is very simple. Here, sodium channel conductance. And normally, when you add mechanism, all sodium channels or any parameters is constant in time and constant in space. It's just constant. But what you can do, for example, you can modify sodium channels conducted in different ways. You can just add stochasticity. We have very big list of stochasticity here, like from white noise to color noise, different type of color noise. And you see it's immediately appear here. It's how it looks like a statistic, but you can just you can just see space distributions, you can just distribution density function, all of things, just play with autocorrelation function, everything, and just immediately see, for example, how the change in the stochasticity, keeping the same number of channels, just only change stochasticity of channel, you can see how change the dynamic of voltage. Okay. It's just example sodium channels, but you can just modify release probability or another channels, or even distributions. This is an example how you modify any parameter in time. You just add, even just the add constant. You can just play and add, modify it in time. And what is the next? Next, you can just modify the space. What I told, when you add parameters, normally it's a constant. This is your question. You can just, for example, here the channels. When you can just add, you can just list of parameters. Here just only parameters, but you can just say list of kinetics and just modify here. It's special table. Okay, and here, the sodium channels, it's a constant from soma to the end. For example, from this to this is constant density.
So all the isoforms, for example, of sodium channels?
What?
All the isoforms of sodium channels could be incorporated?
Oh, yeah. And what you can just do, you can look at just simulate when sodium channels uniform here. Immediately see, you can just play in different ways. How it's, you can just see uniform, linear, like a quadratic, poly. We all just do some experiment. For example, we measure an experiment with the least probability as function of distance, and we can just table, and here the options, you can just in table, just fill this table, and immediately here. This is uniform. You can see here is the action potential. Then, just only example of linear distributions. Okay, now you see, you can just design. You can just keep the same number of channels, only redistribute along the dendritic list, like linear. And see what's happened. It means the number of channels is the same, but because of the maybe diffusion or redistribution somewhere or something, you can see what's happened with immediately action potential here. The close to soma, it's like a zeroth channel, then the distal part is just growing very, just like a maximum here. You see this action potential probability decrease here. But then what you can do, just opposite way. A lot channel close to the soma and small number at the distal part. And it's expected you will see this frequency of action potential really increase. You can see what's happened here. You can just manipulate how to apply. Yeah. Okay. And this is interesting example how you simulate. How you can simulate two cells together. Okay. You can just download. You prepared yesterday astrocyte, save. Then prepare neuron, save. And today you just decide, okay, I need just to see what's happened when just they working together. You first download astrocyte, then you download neuron. Here you see this neuron. Now you can just manipulate where the location, how they locate each other, where this astroglia close to neuron in 3D space. You can just play, and when you're just happy, okay, I'm just happy. And then what can you do? You can design extracellular space. I just move it here. And then, okay, you look here, 10 seconds. 10 seconds, it starts to simulate. What's happened between two cells? You see here immediately mesh here, of diffusion mesh here appear. And normally it's compute very fast. And you will see after the end, you will appear special windows. Because I just decided to check what is the potassium here, but you can just check what other neuron around this after this communication. And you see a lot of potassium around neuron, but close to the astroglia, it's constant. Astroglia, because they have a special channels which uptake potassium, try to keep extracellular space constant. It's normal in vivo. Astroglia keep extracellular space in constant. Yeah.
How do you deal with intersections between the morphologies?
What?
How do you deal with intersections between the morphologies? Like, for example, if you have two neurons and the dendrites are intersecting each other in this.
No. Yeah. It's you can do here because we design extracellular space. They just intersect, but not just touch each other. It means you can even just touch. It's not cross each other. It's still close each other, but not cross each other, because in this case you can only simulate extracellular diffusion. When you put cell, even looks like they just cut or just intersect each other, but it's not the case. Normally they're very close, but you can see the closer you can see this. But it's also.
So do you simulate this in 3D then?
Yes, everything is 3D. Yeah, it's
that's why in the projection.
It's just only here, just only projection.
Yeah.
Here.
Yeah.
But normally you can see different level where it's going here. Yeah.
Can you make some comment about the numerical methods used to solve the extracellular potassium equations, as well as how we can, if it's possible at all to act on them and choose them and modify them?
Could I just suggest, rather than getting straight into questions, we finish the talk and then there will be time for questions.
Just very short. Yeah, you can do it here. Yeah, but these examples are very quick. You can just study not just only two cell. You can just study, like here is the example 9 astrocyte, and one astrocyte here with higher conductance of potassium channels, and you see what's happened when you increase extracellular potassium. You see the dynamic of potassium, it's a different representation here. You see here the concentration of potassium, it's different than here and here. It's this conductance of this astrocyte just try to keep the potassium at the constant layer, compare this small conductant astrocyte. You can just play with different concentration and so on. Okay, it's very short. Also what you can do. And here, the example you can do control release glutamate via uncaging. It's really very complicated experiment with brain cell. This uncaging, you know this. You have just special molecules, two molecules, glutamate and neutral, just bind together. It now is in neutral molecules. You can just put an extracellular field somewhere, and they take a laser, and very small, I don't know, like 1 micrometers diameter, very fast, affect this space. And laser just cut in like a release glutamate. You can like a simulate in vivo or in vitro simulate release glutamate in real like a synaptic. And you can do here, you can just create here like a brain, here spines, here is a release, and you see how it changes concentration here, glutamate, and you can see how it changes concentration around. It means it's very complicated experiment, but with brain cell you can do it very easy. This example, it's probably just few seconds. This example, how we simulate, you can design real axon with Schwann cell. You can just give special activation. And what you can just see here, you can just see how the action potential in different places, what's the potassium concentration close to the membrane, how they propagate action potential, and you can just modify the kinetic of the Schwann cell or axon and see what's happening. For example, you can just see how fast, if something happens with Schwann cell, you will see the action potential decline or go into very excitable state. And this is probably just for information. We design brain cell in such a way, because normally it need a lot of computation. It's very difficult to just sit in front of computer, just wait 1 hour, 2 hours. But what you can do, you design your setup, and then you go export here. Go export. You can just say, I need just to compute frequency of action potential when the release probability change from 0 to 1 for all synapses. This step 0.1. You can just put here everything and then export, okay? And you have just special file, and you can just compute that file on your computer or supercluster. But what I'm just recommend you, this is a free high-performance computer supercluster for neuroscience. It's free for everybody. It means you can just go there, ask open your account, and it's really in San Diego, it's enormous good. And what we did for BRAINCELL, you can save in special format, which exactly for this supercluster. It means you can just save here, create account, and you know, when you just export, you just create only one file. And you know it will be sitting, okay, I will need just to compute this, current, voltage, doesn't matter. When I change this, and this. And when you go on your local computer or cluster, it will create, when next day you will come, and cluster, they send your email, it's done. And you have just like a files. Okay, these files, frequency of action potential at this set of parameters, frequency, and because it's the frequency and release probability stochastic, they create immediately file, mean, statistic, and you just come, you don't need to even analyze data. You just look, okay, good, I like it. Then you can just use all of this big result of computation. And this is really cool for me, because I try, it works perfect for this supercluster. Yeah, this is a simple example how you can just design your task, what is the email you get here, and what is the result you can just download or just look on the web page. I just recommend not only for BRAINCELL, for any neuroscience or computer. They just only check you're just working in neuroscience. You need just to give like an email, university, and so on. Okay, that's done. And I really appreciate these people who work with me about this. This is my PhD student, Cielan. Now she's, she was master student in the design in alien axon. Now she's PhD student. This Professor Rusakov, a lot of communication, discussion, so on. This guy from Ukraine, he a lot of this coding in Python, and really appreciate all of them. But yes, most of them is done by me. If any questions, just ask me. I know everything about this BRAINCELL. And yeah, that's all. And finish. Thank you very much for your attention.
We've got time for.
I have a question. Have you also? I'm guessing you incorporated the calcium dynamics within the cell, like calcium induced, calcium release.
Oh, yeah.
Between the R and the.
Yeah, okay. Yeah. You see, it's like a Lego. When you design calcium release, calcium dynamic, you only just need to change. Okay, I need just put here the calcium induced calcium release, calcium diffusion. You just put here. And immediately you have just all the mechanisms included in whatever you want to simulate, and you have just list of parameters which is known. You can just play here, just modify these parameters, that's all. It means I'll not show just like, we get some result, even just published, especially published result about how to depend dynamic of action potential depending on synaptic release probability. It's very easy. But also we just for use here for point channels dynamic of potassium, and you see sometimes it's like a new software. You need just time to understand, to work, but it's if you don't like something, you go or write your file. It's very easy. Just put your file in special place, then just say, okay, I need just to add to BRAINCELL. It appear. You can just play with it. It's not just like a finish. It's tools.
Did you want to ask Dan?
So we've seen in your own simulations of ODEs in time and also PDEs, like these extracellular potassium concentration that you showed. So what I would like to know is what are the numerical schemes that you use for that?
Yeah, you see, yeah.
Or how much room there is for
no.
changing them.
Yeah, it's a really good question. We introduced here two schemes and describe like two schemes. One more like a general, I don't really like. It's more like a summation of extracellular potassium, like a nonlinear summation. But another, like a typical diffusion, you have like a mesh, you have diffusion coefficient, you have just like a tortuosity parameter, and this part of this scheme was introduced in Heinz from Neuron. We a little adopt here. It looks like a work perfect with some boundary conditions, with I don't know, just it's like a solve. How you say diffusion uptake kinetic equation. You have just like a diffusion plus uptake. You can simply modify this extracellular space. We introduced two schemes. You can just compare. They both looks okay, the same, but sometimes I prefer this scheme with typical mesh, extracellular mesh. It means how it works. You have mesh. You have some cubic. And every membrane inside this cubic, just assume it's like a point, assume the total membrane for release potassium, sodium, so on. And then just simulate. It means we don't need just scale all of this complex neuron in extracellular space. For simulation extracellular diffusion, everything inside this cube, for example, here's the example mesh like a 50-50-50 for 200 micrometers. It's a big. You can just create smaller, but it will increase time of computation.
Okay, I think in the interest of time, we'll leave it there. Thanks again, Leonid.